Hi everyone, and welcome to your first narrated PowerPoint. In this presentation, we're going to learn about cellular biology. So why do we care about cellular biology when we're studying to be nurses? Well, because if we understand what cells do and how they can be negatively impacted, it's going to help us understand all the different diseases that we learn about. And understanding those diseases tells us what we need to do for patients and how we need to intervene. What I mean by that is if you have a really solid understanding of pathophysiology, then you can really think through what's going to happen with the patient and what you need to do to keep bad things from happening or to help them get better. So, for example, if you understand that the plasma membrane its job is to enclose the cell and control movement of substances across the cell and to manage the metabolic function, then you can understand why a disease in that membrane might happen. So an example of that is Duchenne muscular dystrophy, and that's what you see here in the picture. This is actually a genetic disorder that negatively impacts a specific protein called the dystrophin protein. This protein is imperative for maintaining the integrity of our muscle cell membranes. So when we don't have dystrophin, because we've inherited some crazy genetic problem, then the muscle cells can't repair themselves appropriately. And over time, they keep breaking down and breaking down. Your cells do that all the time. Like you lift weights, you get up, you move, you go for a walk, they break, they repair, they break, they repair. It's all great. Well, with these guys, they break, they don't repair. And as a result of that, they end up with progressive muscle weakness and degeneration. Specifically with Duchenne muscular dystrophy, the proteins are supposed to anchor the muscle cells, and those anchors get broken. And then there's no ability for the muscle cells to actually do their job. So you are going to need to refresh your A&P a bit on eukaryotic cells, and we'll go over them and all their components over the next couple of slides. So the nucleus, the main job is cell division and control of the genetic information. When the nucleus is malfunctioned, the genetic information that ends up in the next cell will also be malfunctioning. Ribosomes are RNA protein complexes that provide sites for cellular protein synthesis. Protein is imperative in the production and replication of DNA and all the other things that the cells do for our body. The endoplasmic reticulum, its job is in the folding and transport of proteins and liquid components of most of the organelles, and it also will sense cellular stress. So one of the jobs that the endoplasmic reticulum does is involving the folding and transport of proteins, and we need that, again, in order for our cells to be able to grow and to reproduce, make more of the cells. So when we have problems with folding and transport, that's when we end up with genetic changes or alterations that can actually end up causing a disease or a specific genetic condition. Golgi complex is responsible for processing and packaging proteins into these little secretory vesicles that can then move in and out of the cell to wherever they need to go. The lysosomes are enzymes, and they are going to digest cellular substances down to their base form, like an amino acid or a fatty acid or a sugar. They're important in processes like cellular injury or adapting to some sort of stress or insult because they get released whenever a cell is injured so that a sick or dying cell can actually be identified and then taken out. We don't want to have a body full of a bunch of sick and dying or dead cells, so our body tries to kind of clean things up. Lysosomes also tend to act as signaling hubs for cellular adaptation processes. The perioxomes, sorry, they are similar to lysosomes, but they have a very specific job. They produce or they use hydrogen peroxide, that's where they get their name, and their reactions are used to detoxify cellular waste products. So the mitochondria is going to contain the metabolic machinery that the cell needs in order to make energy, and it makes energy in the form of ATP, and we need that ATP for everything that happens in the body. If you don't remember that sodium potassium pump system very well, please go back and refresh your A&P. If you sold back your A&P book, don't worry, because your patho book contains a lot of that as a refresher in the beginning of each unit. So one example of a condition where the mitochondria is damaged is called Leigh syndrome. Now, I don't want you to memorize Leigh syndrome necessarily because I'm not going to test you on this because it's pretty rare, but I am giving you an example of what happens. So there's a damage to the mitochondria of the cells, and this ends up causing neurological disorder. We usually start seeing this either in an infant or by the time they are a child. And what we see is this progressive kind of loss of mental abilities and movement abilities, because nervous system cells can impact not just your brain, but also can be fed out to give signals to muscles. So that's one of the things that we see are movement problems when we have certain types of nervous system problems. So Leigh syndrome is caused because there's a mutation in the mitochondrial DNA or the nuclear DNA, and that it ends up impacting negatively the mitochondria's ability to function, so that it leads to a deficit in the mitochondria's ability to create energy in the form of ATP. As a result of that, the body is going to try to form energy from wherever it can get it for these particular cells. And as a result of that, it's going to end up using anaerobic types of energy production, which causes a buildup of lactic acid. So the people that have Leigh syndrome tend to get lactic acidemia. When you see that ending emia, it just means in the bloodstream. Okay, so here we're talking about lactic acid building up in the blood. So they end up with too much lactic acid in the blood. They also end up with hypoglycemia. Glyce usually means glucose, hypo, low. So hypoglucose in the blood, right? Not enough glucose in the blood. We need enough blood sugar in just the right amount. We don't want too much, we don't want too little, but we need it in just the right amount for cells and the body to have the nutrients that it needs to make that ATP. So in Leigh syndrome, without the ATP, the body's using different forms of energy production, and the person ends up with lactic acidemia and hypoglycemia, neither one of which are also good for your brain's ability to function. So the cytoskeleton is basically the bone and the muscle of the cell, and it has networks of protein filaments, including things like microtubules and active filaments. And these cytoskeletons also form cell-like extensions like microvilli, cilia, and flagella. And hopefully you remember those from A and P, particularly the cilia, which we see in our respiratory tract, and the microvilli, which we often see in our intestinal tract. These are things that are like little fingers or little hairs that can move fluid or substances out of the way, but they also provide a really good surface area for absorbing. So the microvilli, because they are in the duodenum of the intestinal tract, they provide more of a surface area for nutrient absorption. So when we have something that impacts those little microvilli or the cilia, we can end up with diseases. So one example of that is actually in the lungs, and it's called primary ciliary dyskinesia, or PCD. This is a genetic disorder that gets inherited, and the inherited problem is a defect or an absent cilia in the respiratory tract, and it causes chronic respiratory tract infections. It can even be bad enough that we end up with some abnormal organ positioning, and that may also be another byproduct of the genetic inheritance, and it causes infertility as well. So the impaired cilia in the respiratory tract can't effectively move any mucus or other substances, and this is why stuff kind of gets trapped there, and they end up with these chronic respiratory infections. The plasma membrane encloses the cell, and it controls movement of substances in and out of it. And we really need that plasma membrane to be able to do its job. So if we don't have a functioning plasma membrane, then we're not going to have the substances inside the cell that we need for the cell to work, like potassium and other things. And we won't have a lovely balance between the inside of the cell and the outside of the cell. The plasma membrane exerts a powerful influence on metabolic pathways. It's also going to be involved in cell-to-cell recognition because it's basically the outside of the cell. It's involved with cellular mobility, the cell's ability to move or to go where it needs to go, and it helps to maintain the shape of the cell. And you might think, who cares? You know, a cell is a cell. You'd be wrong. We do care. And one of the biggest examples of problems with the plasma membrane is sickle cell anemia. And over here, I've given you a picture of what a normal red blood cell looks like. It's the one that looks like a gummy lifesaver, right? It's round and kind of concave. It holds lots of hemoglobin so that the red blood cell can carry oxygen to the tissues, and it drops the oxygen off there, and then it picks up all the waste, the CO2, brings it back to the lungs so we can breathe it out. It's imperative for all the cells of our body to get oxygen from our red blood cells. Well, with sickle cell anemia, there's damage to the plasma membrane of those red blood cells. This is actually caused by an inherited genetic mutation in the hemoglobin gene, and it causes a production of an abnormal hemoglobin that is referred to as HbS, and the S stands for, you guessed it, sickle, for sickle cell anemia, right? So the abnormal hemoglobin now causes these red blood cells to become stiff, rigid, and take on this weird sickle shape that you see over here in the picture. And that happens particularly when there are environments where the person is sick, they're dehydrated, or they don't get enough oxygen. So those are things that will trigger this process to happen. And then that plasma membrane, it becomes very damaged. Well, this damaged membrane results in the cells becoming more fragile and breaking apart, which you can see here. And then it causes hemolysis, which is just a fancy way of saying breaking apart the cell. Hemo is the blood cell, and lysis just means breakdown. And then what you see here is as a result of them not being this beautiful, nice little round, pliable blood cell like it's supposed to be, those regular cells can get through small little narrow places like arteries and capillaries and then pop back open on the other side, but the sickle cells can't. So they obstruct the flow of blood, and they cause what's called ischemia, or lack of blood flow to the tissues, which then causes hypoxia, which is another way of saying not enough oxygen getting to the tissues. So they'll basically starve and die. So the plasma membrane involves the lipid bilayer. Hopefully you remember that from A&P. And it has the two layers. You remember the hydrophilic and the hydrophobic layers. Well, that's important because it provides a barrier to the diffusion of hydrophilic substances while simultaneously allowing fat-soluble molecules to easily pass through. So the membrane proteins can extend across that bilayer, be inside of it, or be mainly on one side or the other, or they can be outside altogether, depending on what the protein is. These membrane proteins and the other proteins are going to get synthesized by the ribosome and then get translocated through a process that's called trafficking. If you're laughing at that going, oh, it's kind of like human trafficking, kind of. It's trafficking of stuff inside and outside of the cell so that it can get where it needs to go, so it can trigger a process inside of the cell. So that's what happens. The process moves the protein to a different place inside of the cell, wherever it's needed. In order to be able to do that, the membrane has to be able to take the proteins and fold them and move them, and that requires that the membrane is stable. And when it's not stable, then we have substances that can't get into the cell and can't do their job the way that they should. And that can result in misfolded proteins. So misfolded proteins are the root of many different diseases. And one common example is Alzheimer's disease. This is actually a neurodegenerative disorder in your brain. Neuro means, nervous system, and degenerative means it's falling apart, basically. And what is happening in Alzheimer's is that there's an accumulation of these weird misfolded amyloid beta proteins and tau proteins in the brain. And these misfolded proteins form plaques, which are like stiff little buildups of these things, and they tangle together, and that's going to disrupt the normal cellular function. And what ends up happening is it causes this progressive loss of memory and cognitive abilities. It's very, very sad. So proteins are incredibly important throughout our body, and they're going to be involved in many different diseases and conditions. Proteins determine how well the membrane functions. They act to recognize and bind with other substances to move them inside and out of the cell. They form pores, and they transport channels so that we can move substances and store them appropriately. They produce enzymes that drive those pumps, like the sodium-potassium pump, or they maintain those ion concentrations. They act as cell surface markers, and they have the ability to form cell adhesion molecules so that we can stick together and actually have tissue. And they can even start chemical reactions. So proteins are incredibly important. They form cellular receptors. They have these little receptors on them that recognize and bind with smaller molecules, which are referred to as ligands. So what do those cell surface markers do? Well, 5 different things right here. Cell identification and classification. So basically the cell surface marker acts like a fingerprint and identifies and classifies the cell types so that the body can distinguish between a normal cell, a cancer cell, a stem cell, or an infected cell, because the body is going to react to things like, hopefully, cancer, not always, and infected cells to try to remove them. It's going to respond to stem cells to try to get them to change into the type of cell that is needed, and it's going to respond to normal cells to make them function the way that they're supposed to. Cell surface markers also are involved in cell-to-cell communication. These markers facilitate communication between the cells because they act as kind of a docking station for these little signaling molecules, and once the signaling molecule docks, then it can trigger a cascade of signals inside of the cell and between cells, and this is going to be important to do things like growth, differentiation, which is another word for that means changing into a more mature form, and even migration of cells. And you might be thinking, why would cells migrate? Cut yourself and you will see the process of migration. That is what your body will do to try to heal you whenever you have an injury, right? Even that little paper cut that you get turned in a page. In immune response, these cell surface markers help the immune cells recognize and respond to specific pathogens. For example, T cells use specific markers to identify infected cells, and B cells use specific markers to target the pathogen itself. They also function in cell adhesion and migration. The surface markers help the cells stick together and stick to each other, which we need, again, for tissue formation and wound healing, and to migrate cells to specific locations. And they're also important for drug therapy. So cell surface markers are used to develop certain kinds of medications. So by identifying the marker on a diseased cell, scientists can actually create medications that target that specific cell. Proteostasis is the process of cellular balance of processes. It is the state of balance within a cell during the process of protein synthesis, folding, and dehydration. And the proteostasis network is actually made up of ribosomes that are also known as makers, and chaperones, which are also known as helpers. And they're going to be involved in breaking down proteins or proteolytic systems. And again, lytic means to cut or to break. So the carbohydrates on the outside of the plasma membrane are going to form a coat that actually protects the cell from mechanical damage, and it makes this weirdly slimy surface that helps that cell to move too. And the carbohydrates are also going to function in that process of cell-to-cell recognition, so our body isn't removing cells that we need. That's the end of this little presentation, and next you'll move on to the next one. Make sure that you're jotting down some notes because you will have a quiz at the end of this module before you come to class, and I want to make sure that you understand all of the information.